Welcome back to Bio 111. This is Chapter 16, Section 1, Gene Expression. So how does gene regulation, benefit an organism? Well, first think about eukaryotic cells, multicellular organisms. Each cell in your body has the full genome, but they're not all the same. You have eye cells, brain cells, skin cells, heart cells, all doing very different things. They do different things. They're able to do different things because different genes are turned on in those cells. All right? We don't need them all. And the way these genes are turned on and turned off, we call that regulation. Right? Another reason why it's important, this is true for prokaryotic cells as well, generating protein, transcribing protein from RNA to DNA, RNA to protein, takes a lot of energy. We don't want to do it if we don't have to. We want to be efficient about it, only use it when we really need it. Space. If we're making a ton of protein we don't need, where are we going to put it? Right? We need to be cautious about that too. Cells are only a certain size. And then time. We only have so much capacity to generate proteins. And some proteins need to be created really fast. And if we've got a log jam where we're making one protein that we do not need, the protein we really do need isn't going to be made in time. All right? So just looking at different types of cells, we just mentioned this, all of the cells in your eye, in your liver, your spleen, pancreas, and all the other parts of your body, other tissues of your body, they all have the same genes, all have the exact same DNA. And the reason why they look different, they function different, is because of gene regulation. They all have the same genes, but not all genes are turned on. And different genes are turned on. All right. This is done through genetic regulation, which we're going to be seeing in the next chapter, or in this chapter. All right. So, as is the theme with this class so far, there are generally two contrasting ways in which transcriptional regulation or regulation of gene expression are carried out: prokaryotic and eukaryotic. All right. Remember, prokaryotic are cells that do not have a nucleus or really any other organelle inside of them. They're very simple. In a prokaryote, mRNA is translated and proteins are transcribed simultaneously. There's no separation. There's no nucleus, right? We'll see what this is different when we look at eukaryotes in a little bit. So transcription and translation occur simultaneously in the cytoplasm. There's no nucleus for it to occur in. And regulation occurs at the transcriptional level. Now, eukaryotes are different, right? That's us. Plants, animals, fungi, the big multicellular organisms. Eukaryotic transcription and RNA processing occur inside the nucleus, right? So the DNA is inside the nucleus, if you remember that. We have to go inside the nucleus to transcribe it into RNA. We make our mRNA inside of the nucleus. That mRNA then travels outside the nucleus into the cytoplasm, where it is then translated into protein. Now, this allows for more points of control, more points of regulation in the process. First one, transcription. We can regulate the transcription, whether it gets turned into an mRNA or not. The gene gets transcribed into an mRNA or not. Translation, whether or not that mRNA, even though it exists, if it actually gets translated into a protein. And then post-translational modifications of proteins. We can then, once we have the proteins, we can go in and then modify those proteins, right? We've seen this quite a bit, the contrast between prokaryotes and eukaryotes. Prokaryotes lack a nucleus, eukaryotes have them. In prokaryotes, DNA is found just floating in the cytoplasm. Eukaryotes, the DNA is found inside the nuclear compartment. Prokaryotes, RNA transcription and protein formation occur simultaneously. There's nothing to separate them, as there are in eukaryotes. And again, in eukaryotes, RNA transcription happens inside the nucleus, and translation of the RNA happens inside the cytoplasm. In prokaryotes, gene expression is regulated primarily at transcriptional level, so as it's being transcribed from DNA to RNA very early on. And in eukaryotes, it happens at multiple levels that we just discussed. All right, so that's it for section 1. We'll talk about section 2, prokaryotic gene regulation, in the next video.